So far, we have talked about the important role that is played by the endoplasmic reticulum as the entry point for the synthesis of all the proteins that will be incorporated into membranes in the cell. However, that's not the only important function that is associated with the endoplasmic reticulum. There is another one that is also quite important, and that is to provide a place for the synthesis of all the major phospholipids and all other lipids that are used for the formation of membranes within the cell. Specifically, there are three main types of lipids in membranes. The first one is phospholipids, which are by far the main component of biological membranes. The second component is glycolipids, which are kind of secondary to phospholipids. And finally, there is another lipid present in membranes in mammalian cells, and that is cholesterol. Okay, so we will start by talking about the synthesis of phospholipids, which again, they are the main component of biological membranes in mammalian cells. The synthesis of all phospholipids in mammalian cells takes place in the cytosolic side of the membrane of the ER. But before going into the process itself, it is important to mention that there is one exception to that rule. There is one phospholipid that is not synthesized in the ER, and that is sphingomyelin. This phospholipid is synthesized in the Golgi. The synthesis of all phospholipids within the cell requires two different precursors. The first one is glycerol 3-phosphate, and the second one is fatty acids. Fatty acids are very insoluble in the aqueous environment of the cell. They are hydrophobic in nature, and therefore they need a carrier. And the carrier in the cell is coenzyme A. So, in the presence of membrane-bound enzymes, the fatty acids that are carried by coenzyme A will be linked to glycerol 3-phosphate, and this process drives the insertion of those fatty acids into the membrane of the endoplasmic reticulum, therefore generating phosphatidic acid, which is the first product in this pathway. The phosphate group in phosphatidic acid is then eliminated, and that generates diacylglycerol, which is shown here, and this is the true precursor to all other phospholipids that will be generated within the cell, diacylglycerol. From this moment, the synthesis of all phospholipids will involve the attachment of different chemical groups to the hydroxyl group that is exposed to the cytosolic side of the ER membrane in diacylglycerol. For instance, in this example, if you add choline to that hydroxyl group, you generate phosphatidylcholine. So the addition of the choline group to that hydroxyl, this is the choline group, generates phosphatidylcholine. Similarly, if instead of adding choline, you add ethanolamine to diacylglycerol, then you end up generating phosphatidylethanolamine. Phosphatidylethanolamine can then be used to generate phosphatidylserine, and this requires the substitution of the ethanolamine group by serine. So if you substitute ethanolamine by serine, then you end up with phosphatidylserine. Finally, if rather than adding ethanolamine or choline to diacylglycerol, you add, instead of any of those, if you add inositol to diacylglycerol, then you end up generating phosphatidylinositol, which is the last phospholipid that is generated in the ER. Okay, so all the phospholipids that we have been talking about have been generated only in one of the two sides of the lipid bilayer. That is, they were generated in the side of the ER membrane that faces the cytosol. And over time, the synthesis of new phospholipids is going to generate the situation that is represented here, in which the only growth that you have is in one of the two sides of the lipid bilayer. Now, since this is not ideal for the cell, there must be a mechanism to flip the new phospholipids from one side of the membrane to the other side of the membrane. And that is done by this enzyme, flipase. The reason why flipase is required for this translocation of lipids from one side of the lipid bilayer to the other is simply because of the hydrophilic nature of the head of the phospholipids. It's highly charged and it's very hydrophilic. Therefore, it won't be allowed passage through the hydrophobic environment of the middle of the membrane. In order to flip from one side to the other, you need flipase. Lastly, it is important to mention that it is not only phospholipids that are synthesized in the ER. Cholesterol, one of the other very important lipids within the cell, is also synthesized in the membranes of the endoplasmic reticulum. Similarly, ceramide, which is the precursor to all glycolipids, as well as to sphingomyelin. Sphingomyelin, by the way, is the only phospholipid that is not synthesized in the endoplasmic reticulum. So these two extra lipids, ceramide and cholesterol, are also synthesized in the endoplasmic reticulum. And with this, we have covered the synthesis of lipids in the endoplasmic reticulum.